Thank you. So I am excited to be talking to everyone today. I'm going to be walking through technical considerations in urobiome research, especially the low biomass constraints that may shape study design, extraction, and interpretation. So before I jump in, I just wanted to give a quick introduction so you know a little bit more about me and my background. So I started with a BS in cell biology at Rice University in Houston. And I stayed in Houston to work as a research assistant in an endocrinology lab at Baylor College of Medicine with Dr. Stephanie Sicily. And then from there, I moved to Oregon, where I completed my doctoral work with Dr. Chris Carton in a food microbiology and biological data science program. And this is where I really started to bridge my bench work with my computational analysis work. And currently, I'm a postdoctoral fellow at OHSU in the Nye Cancer Research Building and DMICE, working with Dr. Lisa Karstens, focusing on urobiome research. And the images that I included in my slide are just little snapshots of different experiences and skills and techniques that I've been graciously exposed to over the years. So as the human microbiome field has matured, questions about host microbe interactions have expanded beyond the gut and into other body sites, as well as into how these microbial niches impact human health. And that expansion has really allowed us to think about scale. And what I mean by scale is that there are body sites that are considered high biomass ecosystems where microbial signal is abundant and relatively robust. And then there are low biomass environments where microbial cells are far fewer. And in these settings, there are technical variations that can meaningfully shape what we detect. So the urinary tract is one of these low biomass niches. And the urinary microbiome has been associated with multiple urologic conditions. That being said, the urobiome has not been characterized to the same depth or consistency as high biomass systems. So this is kind of the key premise for what I want to talk about today. I want to talk about the theoretical frameworks that guide experimental design when we ask questions about the urobiome. So this figure is designed to help quantify what I mean a little bit more by biomass. Urine tends to fall on the lower end of the spectrum when we're thinking about bacterial cells per milliliter. And this has important implications. First, there's stochasticity that increases, which means that small differences in how much material is collected during sampling can really shift the community that we observe. Secondly, signal to noise ratio tightens, meaning that background DNA from reagents, the lab environment, or host cells become a much larger proportion of what we ultimately sequence. So culture-based techniques, including enhanced quantitative urine culture, fantastic at confirming organism viability and very useful for enabling downstream functional studies. But culture-dependent methods are inherently going to be limited by growth conditions, and we might not be able to recover all taxa present, which is a limitation when doing exploratory analyses. And this limitation is one of the reasons why 16S rRNA gene sequencing has been so transformative in this space. It's relatively inexpensive, it's scalable, and again, it's culture independent. And I don't think I have to preach to the choir about the boon of 16S rRNA sequencing here. So this approach targets the bacterial 16S rRNA gene, which contains conservative regions shared across bacteria, and then interspersed hypervariable regions, which are short stretches of sequence that differ between taxa and allow us to distinguish and classify organisms. This is where urine presents an interesting technical challenge. Because it is a low biomass sample, the total amount of endogenous microbial DNA may be small, while at the same time, exogenous DNA from kit contamination or host cells might be abundant. So background contamination is going to exist regardless of the bacterial sample. And this figure is intended to illustrate what that sensitivity means in practice. So on the left, we see a no template control. And that's something that should, in theory, in a perfect world, not contain bacterial DNA. But in reality, we don't really see that. We often detect microbial DNA in these controls, and that's because contamination, it happens. These sequences from contamination may originate from reagents, extraction kits, and the general lab environment. And in this ideal setting, the contaminant profile is stable and contained, which means that it can be a reference for filtering background signal across samples. So if we look over now at the middle and the right panels, we're looking at low and high biomass specimen. And again, in theory, both should generate sequencing profiles that reflect their true underlying microbial communities. Biomass alone does not prevent accurate taxonomic assignment. And if contamination is controlled and proportional, both low and high biomass samples should yield representative community structures. This is where things get a little bit hairy or less theoretical. The theoretical model of these perfectly contained scopes do not fully hold up. In plate-based workflows, there's well-to-well spillover, and DNA or amplicons from non-template controls or high biomass samples can transfer into adjacent low biomass wells during library prep and vice versa. So when endogenous microbial DNA is scarce, even small amounts of the spillover can have a meaningful impact and distort the observed community. The resulting profile may then become a composite of true biological signal, reagent and laboratory contaminants, and then exogenous DNA originating from neighborhood high biomass samples. And this is going to disproportionately impact low biomass specimen, which is why low biomass systems such as urine require rigorous contamination controls, careful plate design, and experiment-specific computational decontamination strategies. So sequencing is just one step of a much larger workflow. In low biomass urobiome studies, consequential variation often occurs upstream well before sequencing begins. So from collection and storage to DNA isolation and library prep, all of these stages determine what DNA is even available for amplification. So decisions introduced during all of these stages can have impacts moving forward and cannot be corrected through bioinformatic applications. So going back to where variability can enter, it can enter at every stage of this workflow. Surprise. And I just want to highlight a few moments where we knowingly or unknowingly are making decisions that shape our downstream analyses. So first, I want to draw your attention to the collection stage. So collection methods directly determine what niche you're sampling. So catheterized specimens, for example, more specifically reflect the bladder, while voided urine incorporates urethral and genital contributions. So nomenclature and clarity about sampling location are not just semantic details, but they really define the biological context of the data. And then when we think about storage and handling, delays in cooling or absences in or an absence in preservation can lead to DNA degradation or compositional shifts before the extraction has even begun. And then we're going to move on to DNA isolation. And in this stage, the ultimate goal is to release nucleic acid from the cell, while also removing inhibitors. And so differences in lysis efficacy or inhibitor removal can immediately impact what DNA is even entering the sequencing pipeline. From here, there are further discussions about choosing hypervariable regions and how that impacts taxa that are being amplified. But I really want to pause on DNA isolation for the rest of the talk and just belabor some points about how important that is for downstream. So DNA extraction, like I said earlier, is the first major determinant of what becomes measurable by sequencing. If the cell is not lysed, then DNA is not recovered, and then taxa are not effectively demonstrated in abundance data that we're picking up on. So in that sense, extraction is incredibly imperative, and it defines what is detectable. A challenge to extraction is the microbial cell envelope and how that differs across taxa. So some organisms are readily disrupted while others are more structurally resistant. And because of this variability, no single lysis strategy is universally unbiased. So mechanical, enzymatic, and chemical approaches each differ in how they approach the lysis mechanism. And those differences can produce systematically different community profiles. So there are numerous commercially available extraction kits, and there's no single consensus at the moment on which kit is optimized specifically for urine. So across published urobiome studies, there's substantial heterogeneity in DNA isolation methods across commercial kits as well as custom protocols. So a key point is that many of these methods and kits were originally developed for different sample types, some for high biomass matrices like stools, and others have been adapted for lower biomass clinical specimen. And I'm just going to talk a little bit about DNA extraction and reframing it as two distinct processes. So there's the lysis step, which is how cells are disrupted. And the second step is purification, which is how DNA is recovered and cleaned once it's released. On the lysis side, protocols vary widely. There's enzymatic digestion, bead beating, and I'm going to go into more detail about that soon. And then there's also the DNA collection method, where we can see silica spin columns, magnetic beads, and more traditional approaches like alcohol precipitation in use. And all of these impact DNA recovery, inhibitor carryover, fragment characteristic, and contamination susceptibility. So I'm going to go back to Cell Bio 101 here with this figure and just describe gram-positive and gram-negative envelope architecture in a way that's relevant to DNA extraction, because these differences may predict lysis efficacy. So first, a quick definition. Peptidoglycan is a bacterial cell wall polymer made of alternating sugars that form long glycan chains that are cross-linked by short peptides. And this basically creates a mesh that provides shape and mechanical strength. So on the left, we can see a gram-positive bacteria that has a single cytoplasmic membrane surrounded by a thick multi-layer peptidoglycan wall. that is highly cross-linked. So functionally, this structure can be relatively resistant to chemical disruption alone. And then on the right, we have a gram-negative bacteria, which has an inner membrane and a thin peptidoglycan layer in the periplasm and an outer membrane enriched by lipopolysaccharides. And then once the outer membrane is disrupted, often by detergents, the thin peptidoglycan cell wall is a little bit easier to disrupt. So enzymatic lysis, it's really all about matching enzymes to cell wall chemistry. And in bacteria, the main structural polymer is peptidoglycan. And the alternating sugars that I mentioned are NAG, N-acetylglucosamide, I might be butchering that, and NAM, N-acetyl uramic acid. And NAG carries a lactyl group that then anchors the short peptide stem, which enables cross-linking with mechanical strength. And so with that in mind, there are lysis enzymes that target specific bonds in this matrix. Two common enzymes that act on the glycan backbone are lysozyme and mutinolysine, which disrupt the bond between NAC and NAG in very specific ways. And there are other enzymes that impact cross-links between very specific types of bacteria. And again, there's another bacteria, zymolyse, that is actually disrupting glycans in yeast and fungal cell walls. And so all of this is to belabor the point that the enzyme in a lysis buffer is going to have an impact on the way that a cell wall is being broken down. And so that's something to take into consideration. So broadly, DNA lysis methods, stepping back a little bit, fall into three categories. There's enzymatic, mechanical, and chemical. Enzymatic, I went over on the other side. Mechanical lysis, such as bead beating, grinding, and sonication, utilize physical force to disrupt the cell and can improve recovery of organisms with robust or heavily cross-linked cell walls. And then with chemical methods, we often see detergents or chiotropic agents that solubilize membranes and denature proteins. And again, no single lysis approach works equally well for all bacteria. Up until this point, I've been describing a lot of mechanism, but how do we see this play out empirically when we're looking at bacterial communities in the wild, so to speak? So in this slide, I wanted to just kind of summarize a study that was done by Tung et al. Using a mock oral community. So that's a defined community where the composition is controlled and the benchmark is clear. And on the left is a figure of the workflow of the different lysis methods that were used. And then on the right, there's a comparison of the relative abundance that's looking at these 6 different lysis methods and how the mock community was observed. And so what stands out from these findings was that the best concordance with the expected composition came from methods that combined mechanical disruption with enzymatic lysis. And so that was the bead beating plus enzymatic approaches that are shown here as M5 and M6. So those outperformed enzymatic only or kit only workflows in terms of recovering intended community structure. And so I wanted to move away from like an idealized mock community setting and see how isolation kits work with a sample that we might be using, and that's urine. So in a study that was done by Dr. Carstens and colleagues, they were able to directly test the question that we've been building towards, which is, does DNA isolation method change the urinary microbiome profile you observe? And so in their study, they took 11 voided urine samples, split each sample, and processed the same material in parallel using 5 commercial extraction kits. And the strength of this design is that it aimed to look at primarily the extraction protocol amongst these different urine samples. And so when we look at this table, we can see that there are different combinations of mechanical, enzymatic, and lysis across 5 kits. And so going into the results, so in the left figure, we see DNA concentration recovered from the same voided urine samples across the kits. And you can see clear kit-dependent variability. The blood and tissue kit yields higher total DNA on average, whereas the Promega kit tends to yield lower concentrations. So these results reflect true extracted DNA and not microbial specific DNA. And in urine, we're going to see both host and microbial DNA. So then when we look at the figure on the right, it's important that we look at sequencing depth or total reads obtained. And here the distribution is much more similar across the kits. So in other words, despite differences in DNA yield and concentration, library prep and sequencing produced comparable read counts across the kits, which is consistent with normalization during PCR and library construction. And so the main takeaway that I want to belabor is that kit choice in this instance did not strongly impact total DNA yield, and that it did not automatically translate to differences in sequencing depth. So in prior microbiome benchmarking studies, it has been shown that gram-positive cell wall structure can be a barrier to uniform lysis. And so a reasonable concern is that extraction protocols might bias towards gram-negative taxa. So in this study, the authors tested that directly by comparing the relative abundance of genera of known gram stain characteristics across kits. And overall, 4 of the 5 kits produced broadly comparable gram-positive relative abundances. So there wasn't strong evidence that most kits systematically shifted the community towards a gram-negative organism bias. But this is just a small part of the story of potential kit bias, especially in the urinary niche, where specific genera can be clinically important. So next, the focus was on 8 genera with high biological relevance. 3 of that included common urinary pathogens, and then that 5 other known urinary microorganisms. And so in that analysis, the Promega kit showed a trend towards lower recovery of several gram-positive associated genera, particularly Enterococcus, Corynebacterium, and Staphylococcus, although these differences were reported as non-significant. And then the Power Soil kit appeared to recover more quote-unquote easy to lyse gram-negative organisms like Escherichia and Klebsiella. But it's worth, but the authors, instead I'm reading over my notes. But the authors note that follow-up did not confirm a true gram-negative bias. And so the take-home for this slide was that although most kits didn't dramatically shift gram-positive versus gram-negative overall, kit choice still changes the apparent abundance of specifically clinically relevant genera from these urine samples. So to conclude, the main message that I wanted to show in this presentation is that urine's low microbial mass has an impact on the way that we look at community abundances downstream. And there are all these small considerations that need to be taken into account for reproducible and robust results. And background DNA and play effects can contribute meaningfully, which is why contamination controls are absolutely imperative in the design of your urobiome research question experimental design. And another takeaway is that not all kits are have the same lysis approach. And so when choosing your kit, one, report it, to think about the considerations and the way that the lysis might bias your community profile. And so that is my talk about technical considerations. And if we have time, I'd be happy to answer some questions.
That was really great. It was a very detailed biochemical treatment of this. I really enjoyed seeing that. Um, we're going over on time, but I think it's a very important topic. I'm sure there's some people that might want to ask some questions or have some comments. Does anyone have any questions or comments for Keisha? I see people clapping. It was a very, very clear talk. So in the next step really is kind of thinking about decontamination of these type of data. Do you prefer kind of visual inspection and in thinking about the microbiological plausibility, or a statistical decontamination of the data?
So there's a degree to which it depends upon what step you're introducing contamination in, right?
Mm-hmm.
And there is a level to which you really can't go back. And so if you are at a place where you have your reads, you're sorting through your abundance data, there are different different controls that are put into place to better assess your data. I'm actually going to take a step back real quickly and say that controlling for contamination, there are points when setting up your study design and prior to sequencing where you can lay the stepping stones to more readily determine what is a likely contaminant. So for instance, there are strategies that you can use with like serial dilutions. Serially diluting your mock community so that it becomes more apparent what is likely to be a true source of a contamination versus something that is like fairly spurious and an artifact of spillover.
Mm-hmm.
There are several different methods to approach observing contaminants or trying to identify contaminants, and it can be based off of just looking at your non-template control, having a relative abundance cutoff, but there are some great papers. Dr. Carson's wrote one that
Yes.
have that have looked at decontam methods, and there's a Bayesian approach. I forget what the name of that software program is that has been found to be pretty effective. But these approaches are also tied into how you're setting up your plate at the beginning. Like the idea is to expect contamination and set your plate so that you have these controls to be able to better delineate what is likely to be contamination.
Yeah.
Oh,
Well,
wait.
I do see one hand raised from Scott DeSantos.
Hello, Dr. Harrison. That was a really cool talk. Thanks for giving that. It's always cool to see people talk about contamination, microbial biomass. Um, Quick question for you: in terms of kind of when you've got the kind of table of counts and you're analyzing your data, I know a lot of people like to use decontam in R. Are there any other kind of software packages that you've come across that you'd recommend for looking at identifying contaminants in your data set?
So Audra was very kind to remind me of the name. So what I recommend, because I just read it recently, it was fantastic. There is a paper that was published, I think, in 2019, that was investigating different ways of gauging, delineating contamination, and it did a comparison of decontam and source tracker, and that might be worth reading, to see if that fits your like experimental research design.
Awesome.
Mm,
Thank you.
You're welcome.
awesome. Well, this has been an excellent conversation. I think very relevant for this community, one that we'll probably revisit again and again as we refine our methodologies. Thank you, Keisha.
Thank you for having me.
All right, everyone. Thank you all for joining us. It's good to see you all again, and we'll see you all again at the next one.